Our vision is to be a leading pharmaceutical company in India and to become a significant global player by providing high quality, affordable, and innovative solutions in medicine and treatment. Our competence is to develop, manufacture, and market innovative, affordable medical and health solutions. As far as the company profile is concerned, this company was founded in 1951 with 5 manufacturing plants with more than 7,000 plus employees. We are operating in 100 countries with more than 900 brands and 1,400 SKUs and amongst 152 DMFs. We are operating in 100 countries with 7 regions with sales force of 300 and 200 plus products. With this, I'll not take much of your time and just play a small movie to introduce the company's signature.
Behold, the story of victory, of life over malady. Behold the saga of power, of science over sickness. Behold the tale of triumph, of dedication over disease. Behold the rise and rise of Cadila Pharmaceuticals Limited, one of India's largest privately held pharmaceutical companies based out of Ahmedabad, Gujarat, India. Cadila Pharmaceuticals was born out of the vision of a legend who left an indelible mark in the chronicles of Indian pharmaceuticals industry. He was Indravadan Ambalal Modi, the founder chairman of the company, rightfully christened as the Medicine Man of India. For Shri Modi, healthcare was not a privilege of the few. It's the fundamental right of all.
In 1951, I know that when pharmaceutical industry was not existing in this country and people felt that being a technology-oriented industry, perhaps it cannot be started in this one. And at that time, I decided that in the interest of the country and everything, better we may give a foundation so that other people also follow for this industry.
His vision to alleviate pain of the underprivileged through research-driven innovations has been driving Cadila Pharmaceuticals for more than 6 decades now. Innovations which are backed by the key strengths of the company, the state-of-the-art manufacturing and research facilities. The flagship manufacturing facility at Dholka, Gujarat, sprawls over 100 acres. This avant-garde facility is US FDA approved, besides meeting most of the stringent quality standards across the globe. It manufactures tablets, capsules, injectables, liquids, and orals. The second formulations manufacturing facility is located at Samba in the state of Jammu and Kashmir. The API facilities at Ankleshwar, Gujarat, manufacture a large number of key APIs and intermediates at this US FDA and ISO 9002 and ISO 14001 accredited plant. The company also has set up its first ever overseas formulation manufacturing facility at Addis Ababa in Ethiopia. This world-class facility is the only WHO CGMP compliant manufacturing facility in the region. Cadila Pharmaceuticals offers an ensemble of formulations. The company's extensive range of finished dosage formulations covers more than 850 products spread across 12 specialties with many of India's top brands in its basket. Great goals are defined by far-sighted vision and achieved by seamless teamwork. A multicultural, multilingual, and multinational workforce of over 7,000 employees is truly the propelling power of the company. The company is a significant player in domestic market with a field force of over 4,000 and covering over 250,000 doctors, including all specialists and generalists. Not just in the domestic market, the company has a wide global footprint with over 1,000 marketing authorizations across 85 countries. Its international sales force of over 150 highly diversified and versatile personnel lends it an outreach that blankets the globe. Evolution through innovation is Cadila Pharmaceuticals' credo. As the company grows, so does its commitment to research. Manned by more than 350 scientists, the R&D facilities of Cadila Pharmaceuticals at Dholka, Gujarat is an institution of breakthrough innovations, such as Polycap, the world's first polypill for cardiovascular diseases; Rizorin, the world's first boosted rifampicin for tuberculosis; Mycidac-C, the world's first active immunotherapy for lung cancer; and novel formulations like Rabiloc IV, the world's first rabeprazole in IV form. To take the focus on innovation to the next level, Cadila Pharmaceuticals believes in forging scientific collaborations with world-class institutions sharing the same passion for healthcare research. It partners with leading international companies to discover newer treatments in the areas of novel vaccines, better antibiotics, respiratory diseases, gastrointestinal diseases, cardiovascular ailments, diabetes, cancer, sepsis, and regenerative medicine. The Cadila Pharmaceuticals Group is gigantic. Its group companies complement and expand the dynamic vision of the company across new avenues of healthcare and newer fast-paced business areas. Cadila Pharmaceuticals is a gentle giant. It undertakes a number of targeted activities that are bringing about long-lasting and deep-rooted changes, especially in the fields of health, education, women empowerment, and child care. As a responsible corporate citizen, Cadila undertakes a number of CSR activities in these areas. When the beacon of exemplary leadership passed on from the illustrious Doyen Shri I.A. Modi to Dr. Rajiv Modi, Cadila Pharmaceuticals scripted a new success story in its journey of fast-paced growth phase.
For me, it's a mission, and I have a very simple mission, which is to take this company forward at all costs, to make it into a globally sized business, which will continue to provide the world's best quality, affordable medicines for people around the world, and would strive to bring innovations.
Strategic global expansions and research into areas of unmet medical needs are the future. Decades of dedicated research, groundbreaking healthcare innovations, and worldwide impact proves the infinite potential of one powerful emotion: care. Care that transforms into a driving force for a revolutionary change. Care that translates into affordable miracles for those who need it the most. The success of Cadila Pharmaceuticals bears testimony to the fact that the care continues.
With this vision, coming to the today's topic, which is on a very interesting topic, that is optimizing clinical use of novel carbapenems. For this, I would like to introduce our speaker, Dr. Rajat Agrawal, who is an additional director of critical care medicine, Fortis Escorts Hearst Institute, New Delhi. He is in charge of ICU at Fortis CDOC, New Delhi. He heads 50-bed ICU with team of 16 doctors, member of Executive Committee of ISSM Delhi Chapter 2022, teacher of DNB Critical Care Medicine and ISSM courses, in charge of ICU program at FHI, head of COVID task force at the hospitals in his areas interest are cardiac critical care and antimicrobial stewardship. Also would like to introduce the panelist for the session, Dr. Nutu Jain. She is a consultant PSRI Institute of Pulmonary Critical Care and Sleep Medicine at Pushpati Singhania Hospital and Research Institute, New Delhi. She has done Bachelor of Medicine and Bachelor of Surgery from Government Medical College, Nagpur, DNB Medicine from Batra Hospital and Medical Research Center, New Delhi, and Fellowship of American College of Chest Physicians, USA. She has a great interest and vast experience into critical care, including CVP placement, percutaneous tracheotomy, and managing all critical and mechanical ventilated patients, ultrasound guided procedures in ICU and pulmonology, basic and advanced cardiac life support, bronchoscopic procedures like BAL, TBLB, and TBNA. The second panelist for the session is Dr. Dikshit Kumar Thakur, who is a consultant pulmonary critical care and sleep medicine at RTMS Light Hospital, NFC Delhi. Dr. Dikshit Kumar Thakur is a consultant in pulmonary critical care and sleep medicine. He has more than 10 years of experience and has done his specialization in pulmonary medicine. After completing his specialization, he has done a fellowship in critical care from Indraprasth Apollo Hospital. He has received a fellowship in sleep medicine from prestigious Indian Sleep Disorder Association. He has a special interest in interventional pulmonology. He is running the Department of Pulmonary Critical Care and Sleep Medicine at Fortis CDOC Hospital and treating lung diseases, diagnostics like bronchoscopy, EBURS, ICD, pleural aspiration, ETC, and therapeutics, along with sleep disorders and managing the critical patients in ICU. So with this, I will invite now Dr. Rajat Agarwal to start the scientific session.
Thank you, Mr. Bharat, and good evening to all. So we are a bit late in starting, so I won't be taking too much time. So the idea for today's meeting is focusing on relatively a newer molecule which has come. I would not say it's a totally new molecule, but yes, relatively newer carbapenem which has come in the market. It's called Biopenem. So I'll be briefly discussing about how we can place Biopenem in our current armamentarium in managing the critically ill patients. And we have two good panelists, excellent panelists who will be taking their viewpoints, and then we'll be trying to make a strategy that where we can place this molecule. So we all know that the prevalence of antimicrobial resistance and nosocomial isolates is increasing. This is a no-brainer, and everybody is facing this problem day in and day out. And this is a major concern to all clinicians as resistant bacterial strains can be associated with high mortality rates. This is an interesting slide which we can see here, that as soon as the antibiotics were discovered, like from 1930s, like sulfonamides before the penicillin came from Dr. Fleming. And as the downside from that, you can see that as soon as the antibiotics were discovered, their resistance was also observed within 10 to 20 years of their development. So you can see the sulfonides, resistance started developing in 1940s and 50s. That the tetracycline resistance started developing in 1950s and 60s and 70s. And then we have the carbapenems, which came in 1990s, and subsequently the resistance to that also started developing in early 2000. So this is an unfortunate part. And I would say that we as clinicians are also partially to blame for that because of a rampant use of antimicrobial agents. Now, this is a study which probably everyone has gone through it, and it's published by Vincent et al. In JAMA in 2009. So basically, it was a prevalence study to know what was the incidence of resistant pattern in international ICUs. So it's a big cohort of around close to 14,000 patients. This was the study from which it was a all over the world global study. And this way found out that gram-negative pathogens were common cause in Asia as well as compared to US and Europe. So they had EPIC 1. The second study was EPIC 2. So they clearly showed that gram-negative bacteria like E. coli, Enterobacter, Klebsiella, Pseudomonas, etc., and especially the ESBL producing, the highest incidence is found in Asia. And mind you, this study was published almost 13 years back. Now the incidence of resistant ESBLs and KPCs is much higher than what was published at that point of time. So subsequently in the study, they said the 70% of the infected patients are positive cultures, and the 31% of gram-negative cultures were Pseudomonas. So from this, this was a point prevalence study. So they showed that the majority of infections were gram-negative, and Pseudomonas was the most common infection at that point of time. So I'll just highlight a brief case which we encountered in our practice. This is a case of July, only last month. So we had a 68-year-old male. He is a known case of type 2 diabetes mellitus with severe LV dysfunction. He was an Afghani national. He had a history of benign prostatic hypertrophy. and he was on home Foley's catheter. And the reason was that because he had multiple episodes of left ventricular failure, he was a poor candidate. So the anesthesia team refused to take him off for surgery. And then he was put on, he developed retention of urine, and he had, because of the severe BPH. And then he was, with these complaints, he was admitted to us with dyspnea, altered mentation, and low urine output for past 3 to 4 days. He had the similar episodes 3 months back, for which he was admitted 10 days for 10 days and treated subsequently for that. So on investigations, it was found that he had a leukocytosis, hyponatremia, slightly high creatinine, procal was elevated, and yes, he has a case of severe LV dysfunction. So ejection fraction was only 25%. Poorly controlled diabetes mellitus, so HbA1c was 8.5. So this patient we managed as a clearly case of septic shock. Fluids, antimicrobial agents, cultures were sent. He was started on noradrenaline and other supportive medications, and started on piperacin tazobactam initially in this patient, suspected complicated UTI. So the ultrasound subsequently showed the patient having, you can see the thick wall here in the, it's here. So this is clearly showing that patient has a history of recurrent episodes of cystitis, and then he developed chronic cystitis as a result of that. This was the culture which was found that this was the urine culture which came. If you can see, yes, that it was positive for E. coli. Yes, it was sensitive to piperacin tazobactam. But if you can notice the MIC values, it is almost equivalent to 4. Now, if it is a classically, it's a non-ESBL bug, then the sensitivity has to be much less than 0.5 to 1. But here the MIC was 4, which was borderline going into the resistance pattern. So we can say it was intermediate sensitive. The blood culture also was having a similar pattern. Again, we can see the if you can see here the arrow that the piptazo sensitivity was close to 4. And the other it was sensitive to other antimicrobial agents as well. However, the patient's condition did not improve even after 48 hours of piptazo, and hence the antibiotic was changed to Carbepenem initially. Patient improved after 5 days of therapy, and when they were shifted out of the ICU and then discharged on day 10. So total he received 8 days of injection Carbepenem, and then he was discharged successfully on after 10 days of hospital stay. So this is a brief case which we showed that even the culture was positive. It was sensitive to piperacin tazobactam as per the report, but the MIC was high. So probably the choice of our antimicrobial agent initially was not appropriate in this patient, that piperacin tazobactam. And this patient ideally probably should have been started either on Carbepenem right away, looking into the history of recurrent UTIs and looking into age and comorbidities of this patient. Now we know that last few years we have seen that there's an impact. What is the impact of MDR gram-negative organisms? So approximately 10% of hospitalizations are complicated by healthcare-associated infections. And we know that there's been a recent dramatic increase in the prevalence of resistant gram-negative bacteria like ESBLs, which we just showed a case of, Carbepenem-resistant Enterobacteriaceae, MDR Pseudomonas, and MDR Enterobacter baumannii. And yes, it's a very important aspect which we all take into consideration, that if the therapy is inadequate and not timely, it leads to high mortality. So as we can see here, the adequate therapy, the mortality was almost 16% to 15%, and inadequate therapy or delayed therapy, the mortality was very high, more than 90%. Carbapenems, we all know, play a critically role in our antibiotic armamentarium, and they possess the broadest spectrum of activity and greatest potency against gram-positive and gram-negative bacteria. Due to ESBLs producing organisms and increased resistance challenges present by the beta-lactamases and KPCs or NDMs, Carbapenem still assume a greater role in the treatment of serious infections. So now, is there any really need for a newer Carbapenem? So high mortality and morbidity due to lower respiratory tract infections, complicated UTIs, polymicrobial pathogens such as mixture of gram-positive, gram-negative, and anaerobic organisms, inadequate initial treatment in the increased clinical failure rates and mortality. And Pseudomonas continues to be the most prevalent organism, especially in cases like intra-abdominal infections and complicated UTIs. So the early Carbapenems like imipenem are not able to stable for hydrolysis. That's why we combine them with molecule called DHP reductase called cilastatin, and a co-administered with DHP1 inhibitor, that is a cilastatin. Something about a relative newer molecule called Carbepenem. So basically, it's a novel parenteral broad spectrum carbapenem, primarily used for the treatment of infections like sepsis, lower UTI, lower respiratory tract infections, intra-abdominal and genital infections. So it was discovered in basically Japan, and then its use is quite prevalent in Japan, Thailand, and China since last 20 years. Recently, it has come up in approved by in India, and it has been approved in India so far for the management of complicated urinary tract infections only last year. Now, how is it different from the imipenem and meropenem? So as we can see here, if we can see the structure here, the biopenem has a special thioether substituent which increases the stability towards MBLs, which are implicated in carbapenem resistance. So it is hypothesized, though well, it's not proven as yet, but it is hypothesized that in the patients, sometimes if we have this thioether substituent, it might have a resistance towards MBL and better efficacy in countering the MBL resistance. And unlike imipenem, biopenem has a 1-methyl substitute at C1 position, as we can see here, conferring stability to hydrolysis by renal DHP1. So what it means basically is that it need not be combined with cilastatin, and the full dose can be given because it is not hydrolyzed by this enzyme called renal dihydroperoxidase. Advantage, though, does it has any advantage over other carbapenems? So it is not a substrate of efflux pump. So we know that bacteria resistance is produced by this, one of the mechanisms to produce resistance, and one of the mechanism is efflux pump. That is, bacteria throws out the antimicrobial molecule from inside, and thus it causes the resistance. Now, it is not a substrate for efflux pump because biopenem does not act on that pathway. So Pseudomonas and Astrobacter infections are more sensitive to biopenem compared to imipenem or meropenem. It has a stability in the presence of renal brush. This we already covered, and it has a low convulsive potential, which reduces the risk to patients. So compared to imipenem cilastatin, which is prone to develop seizures, this molecule has lower tendency for that. So it is quite safe in patients with seizures. The spectrum of activity is quite similar to other carbapenems. Basically, it has some gram-positive coverage like Streptococcus and some species of staph. Please, it's only MSSA, not MRSA. And gram-negative bacteria like E. coli, H. influenzae, Klebsiella, Neisseria, etc., all are sensitive to biopenem, as we have the sensitivity of imipenem and meropenem to these bacteria. Now, what about the PK/PD parameters? These are very important when we consider any carbapenem. So as we can see here, the dose is usually 300 milligram. If we compare to 500 milligram of meropenem and 500 milligram imipenem, the half-life is 1 hour. It is almost similar, but the protein binding, as we can see, is quite low of biopenem compared to imipenem and meropenem. The drugs that are minimally protein bound penetrate tissues better than those who are highly bound. So especially like urinary tract infections and intra-abdominal infections, this molecule may have a better role, especially in like infections with Pseudomonas and Astrobacter compared to imipenem and meropenem. So as I mentioned, it is approved by DGCI for complicated UTI infections. But however, internationally, the molecule where it was discovered, it was used as sepsis, septic shock, LRTIs, a second infection of chronic respiratory lesions, complicated UTIs like pyelonephritis, intra-abdominal infections like peritonitis, and uterine inflations and infections, etc. So it has other role besides urinary tract infections as well. The dosage is 300 milligram. It's available as a vial of 300 milligram. It has to be given twice daily, and an infusion of 30 to 60 minutes. And it is available as a 300 milligram vial and diluted in 100 ml saline. And dose can be adjusted according to the age, but should not exceed more than 1.2 grams per day. Monitoring is, yes, contraindications. It is allergic to that. And in hemodialysis patients, it has to be administered once daily. Now, what is the evidence behind the use of biopenem? So this was on this study, the molecule was approved. So this was a phase 3 clinical trial in Indian patients. The patients were around 163 compared with imipenem. Both had 80 patients each. It was done in 13 centers, and the endpoints were clinical and microbiological response. So as we can see here, it was the biopenem is non-inferior to imipenem and comparatively equal cure rate observed in biopenem. So basically, it was a non-inferiority trial. That means the design of the study was to evaluate whether it was non-inferior to imipenem in terms of cure rate and in terms of safety. It was found to be non-inferior in terms of cure rate, and safety-wise, it was also same. Similar study was compared with meropenem, and then it was found to be equally efficacious to meropenem. So, and safety profile was similar to comparable to meropenem. So this was a randomized controlled trial of eight RCTs, meta-analysis of 8 RCTs, close to 1,600 patients. And the key highlights of this was that the Carbepenem at least had a similar clinical efficacy and macrobiological success compared to imipenem and meropenem. It has a high safety and well tolerated by the adult patients. It may be a valuable option for empirical therapy in LRTIs, complicated UTIs, and complicated intra-abdominal infections. And it is found to be, this was an important point, that it was found to be more potent than imipenem against Pseudomonas aeruginosa because of the different mechanism of resistance, which I just mentioned. Again, this was a study published recently, a couple of years back. It was a review of meta-analysis and was found compared to meropenem in urinary tract infections, Carbepenem had a lower rates of side effects, and so, it was a good choice for treatment of complicated UTIs. For pneumonias also, it was found to be effective for initial treatment of severe CAP in elderly patients with chronic respiratory diseases. So again, these were the 2 trials which showed that compared to imipenem and meropenem, Carbepenem is either better or equivalent in terms of potency. Now, this was a study which showed that Carbepenem had a greater bactericidal effect than other carbapenems against Pseudomonas. So as we can see here, all 4 carbapenems are listed here: Carbepenem, meropenem, imipenem, and ertapenem. And we can see the MICs of the Carbepenem is least for Pseudomonas infections aeruginosa, and also it has a good post-antibiotic effect and good biofilm penetration against Pseudomonas strains. So Pseudomonas is quite notorious for developing biofilms. So Carbepenem is quite effective in penetrating that biofilms and treatment of Pseudomonas strains compared to imipenem and meropenem. The MICs were quite low for Carbepenem for Pseudomonas aeruginosa. This was another study which showed that compared to other antibiotics like colistin, fosfomycin, etc., the MICs were quite low for Carbepenem. Now, this was again the point which I mentioned, that degradation by dihydroperoxidase compared to imipenem, it was almost 100% hydrolyzed. So that's why we had cilastatin with the molecule. Meropenem is almost 58% hydrolyzed by this enzyme. Carbepenem, if you can see, it is hydrolyzed, but the response is only 10%. So it is quite free and not affected by this dihydroxypeptidase and can be given easily. With regards to Klebsiella, E. coli, and Estobacter, Carbepenem is showing more sensitivity than other carbapenems in Indian patients. So this study, subsequent to that, showed the MIC's value were quite less if we compared Carbepenem compared to imipenem and meropenem. So the special features are: it has a great bactericidal efficacy compared to for other carbapenems against Pseudomonas aeruginosa, greater bactericidal effect than carbapenems against Estobacter, established clinical efficacy and success rates compared to imipenem and meropenem, more stable than imipenem meropenem for DHP1, improved tissue penetration due to low protein binding, lesser risk of seizures, a good post-antibiotic effect, and low risk of drug-drug interactions compared to meropenem. This for the renal failure. In this study, they showed that if the creatinine clearance is less than 25, the dose has to be given 300 milligram in 24 hours. And if it is 25 to 50, as we can see here, it is 0.5 to 1 gram over 24 hours. So the dose has to be modified for all carbapenems, and this is for Carbepenem. This is the usual dose modification as per the creatinine clearance. So it's a novel parenteral carbapenem recently approved. It extended a greater bactericidal effect than carbapenems against Pseudomonas. In vitro effective biopenem suggests that drug could be a new therapeutic option compared to imipenem producing carbapenemase, and it has shown clinical and bacteriological efficacy in the treatment of wide range of serious infections in adults and children, which is at least comparable with that of other currently available options. So it is a class of carbapenem with efficacy equivalent to other molecules. Advantage of the BD dosing compared to 6-hourly or TDS dosing of other carbapenems. The cost-wise, that's why it may be beneficial for the patients, and has a good efficacy in patients of complicated UTI though the indications for this might be studied further and might enroll to include pneumonias and complicated lower respiratory tract infections as well. Thank you. So I would like to invite our esteemed panelists for their suggestions. Can come here, or you can, yeah, please. So first of all, Dr. Dikshit, I'd like to start with you. Like in patients of complicated UTI, what do you encounter in your practice, the common microorganisms, which are isolated with you? Right. So that's what I think the experience with everybody who's encountering that. Dr. Jain, how is the incidence of ESBLs you see in your intensive care practice?
Instance PSBL is increasing in our practice, and it is close to 60 to 70% in our ICU.
Right.
And studies have also shown that incidence is now from somewhere from 30% to 70% ICUs all across the world. So incidence is definitely increasing.
Right. Yeah. So I think that a problem which we all are encountering in terms of increased resistance, and the traditional BL-BLI combinations are usually not working anymore. Yes, that's why the focus is shifting towards the role of carbapenems. Dr. Dikshit, what do you say about the seizure potential for imipenem cilastatin? I mean, does it for you, is it a big side effect, or how much you have seen in your practice?
Experience, I am more towards the Carbepenem because of this season potential. Although I never have faced it, but my 99% of the times I'm going towards the Carbepenem.
Okay.
Personal experience I'm not having with them.
Achha. Dr. Jain, would you like to comment on that?
We have been using imipenem a lot for abdominal infection in our ICU. Lot in patients who have pancreatitis with a secondary infection.
Sure.
And we have not seen many patients getting seizure. Overall incidence of seizure is somewhere around 1.5% or so with the imipenem cilastatin, and more in patients with renal insufficiency or other complicating factor.
Right.
Otherwise, in my own practice, I have not seen a single patient who had seizure because of imipenem. Though a lot of seizures were attributed to imipenem, but patient did have other causes.
Yeah.
Which could have been the cause of seizure.
So I think I agree on you. So sometimes in our practice also, we sometimes patients on carbapenem develop seizures. Now it is very difficult to pinpoint whether exactly they're because of a drug induced, any metabolic factors, any underlying neurological issues. So yes, hypocalcemia, hyponatremia, they are very prone to the patient develop seizures. But yes, per se, the imipenem as suggested that the seizure potential is not that high, but yes, if it is there, and if we have some other molecule which we can counteract with, probably it may be a better option to treat with that. Another question I would like to ask you, if you have any personal experience with Carbepenem you have used, and how do you, with looking into the evidence, how would you place Carbepenem in your practice? Would you look at it's a good option or not?
Our armamentarium of antibiotic is getting limited day by day, so I'm sure that it will find a place in our practice while we are looking at a lot of resistance bug, but it is difficult to say at this point in time whether it is going to work for carbapenem, other carbapenem resistant organism. Only time will show, and the resistance pattern will be apparent once we have the test to test it against. I have not used carbapenem in any of the patient, and maybe for the patients who are prone to seizures, we might use carbapenem because it's supposed to have lower seizure potential as compared to imipenem and cilastatin, and it is easily hydrolyzed as compared to imipenem cilastatin. So bioavailability of drug can be more than the usual carbapenem. So only time will tell once we use it more often. And as of now, it is only approved for complicated UTI, not for other infections, though it has been used in Japan for many other infections, and it has good lung penetration as compared to other carbapenem. Maybe it's a molecule which we'll use in lower respiratory tract infection or hospital-acquired pneumonias in our patients.
Right. Dr. Dikshit, your comment on that? Any?
I have used it in last few months.
Okay.
So for me, this drug is more of a reserve drug because I run a secondary care ICU and the resistance, the percentage of resistance which I am getting is very less compared to a tertiary care ICU. So I use it when I find that this is a complicated case and when the cultures are also going towards the resistant pattern, or if the patient is in septic shock and there are the high chances of the high risk cases, let's say use of antibiotics in last 3 months. And if he is fitting into a high risk category with a critically ill patient, only then I am using it as a reserve drug of in place of basic carbapenems, but not routinely.
Right. So I think both the panelists have made a very valid point. So it is not a Carbepenem, I would say still in my personal opinion, it's not any wonder drug that it's going to replace polymyxins or cholesterins, nothing like that. The spectrum wise, it is almost similar to other carbapenems. Or what I feel personally maybe is that, yes, it has a good action against Pseudomonas, suggested in various trials. So yes, it's a good option in that. It has been approved so far in complicated UTI and pyelonephritis. We have used in close to more than 15 patients in complicated UTI pyelonephritis with decent results. But yes, the other indications are yet to be explored. The important thing is that does not penetrate the blood-brain barrier as of now. So I don't think it can be used for bacterial meningitis. I'm not aware of the studies of that. There might be some data on that, but still it is not approved so far. So for complicated UTIs with twice daily dosing, the cost factor also comes down for the patient. So it may be a good option, viable option for use in these subset of patients. And yes, as I think the panelists also mentioned that the seizure potential is quite less with this molecule. So if we are using imipenem for say, imipenem basically we are also using for intra-abdominal infections because it has been hypothesized that it is better compared to meropenem for intra-abdominal infections. Though the head-to-head trials, there are some studies on that, but it's not very robust. So and it is not officially approved as of now, but yes, traditionally that has been the practice so far. So Carbepenem may be considered in those subset of patients for usage. And then again, as we start using the molecule further, then we'll get more experience, more data will be generated, and then we can have a further discussion on that. So
it can be useful in patients who have renal insufficiency because elastin is one
yeah.
part of imipenem which is not good for patients with renal insufficiency. So maybe it will replace imipenem elastin in those patients, but as you said rightly, the time will tell.
Yeah. So yeah, still we have some data to collect on that, but yeah, whatever indications are, it's a decent molecule which we have used so far. And the cost sometimes, especially in the patients in private setups, it's always a challenge, like with meropenem and all. So it is maybe considered in those settings and easy to use. So probably, as Dr. Dikshit maybe say, like home ICU settings and like so maybe twice daily dosing, maybe it's easier to use. Like instead of giving a molecule 6 hourly or 8 hourly, so if you give it twice daily, it may be considered in those settings as well. So any comments from the audience and anybody, any suggestions what anybody would like to give? Or they have used, anybody has used it, so they want to share their experience. Okay, so that's it from my side. If there are no questions, then we can like to invite Mr. Bharat.
Thank you so much, sir, for the wonderful session. And I thanks on behalf of Cadila Pharmaceuticals for sparing your time for the event today. And with this, I'll invite our sales head, Mr. Vijender Raika, to facilitate Dr. Rajat Agarwal with a memento. Thank you so much, sir, for giving us your precious time. As well, I'll request Dr. Rajat to facilitate Dr. Neetu Jain. Thank you so much, ma'am, for sparing your time for the session today. I'll also request Dr. Rajat to facilitate Dr. Dikshit Kumar Thakur for being there in the panel and sharing his feedbacks on the molecule. Thank you so much, Doctor. With this, I'll request Mr. Vijender Raika to give a thank you note for today's session.
Good evening once again. I am Vijender Raika based at Delhi. And on behalf of Kerala Pharmaceutical, I would like to thank Dr. Rajat Agarwal for giving us such a wonderful insight on this novel molecule, Carbepenem. And along with Dr. Rajat Agarwal, I would like to say thanks to Dr. Neetu Jain and Dr. Thakur for giving your insights on the carbapenems and your experience on different molecules. So on behalf of Kerala Pharmaceutical, I would like to thank all the medical facilities who are available over here. And I would like to request all of you, please join us for the dinner and drinks. Thank you very much.